Beyond the conventional DMARDs, there is a whole set of biologic agents that also act as disease-modifying drugs in the treatment of autoimmune conditions like rheumatoid arthritis. These biologic agents fit into one of two categories: TNF alpha inhibitors or inhibitors of other cytokines. Before I start talking about the individual agents, it's worth reviewing some basic facts about these biologic drugs. The majority of these biologic agents are what are known as monoclonal antibodies, which means that they are antibodies targeting one specific target or a mono target. Antibodies can be made in a number of ways, including hybridoma technology, molecular biology techniques, or chimeric or humanized antibody generation. It's not as important that you understand how the antibodies are produced as it is to understand the naming convention of antibodies so you can determine whether an antibody is a mouse-human chimera, humanized, or fully human. If you see the suffix imab, that is a mouse-human chimera. A humanized antibody has the suffix zumab with a Z, and a fully human antibody has the suffix umab. Finally, there's also the suffix sept, which indicates a fusion protein. Typically, this is not a full antibody, but does contain a fragment or a portion of a human antibody. When it comes to biologic agents, there is typically only one approved biologic. Any other products that are similar but not identical are considered biosimilar. This is not the same as bioequivalence with small drug molecules, and so it's worth reviewing the definitions of biologic and biosimilar. A biologic is an approved product composed of proteins, nucleic acids, or some combination of those, or a living entity, and that's produced by biotechnology methods. A biosimilar is a biologic product developed such that there are no clinical meaningful differences between the original biologic and this new molecule. However, they are not identical, and therefore the correct term to use is biosimilar. It might be helpful to take a moment to review the definitions of biologic and biosimilar. The first and largest group of biologic DMARDs are the TNF alpha inhibitors, also known as anti-TNF alpha antibodies. The reason TNF alpha is such an important and central molecule targeted by drugs is that it's involved in several autoimmune conditions. TNF alpha is released by many immune cells like T cells and macrophages, among others. And the release of TNF alpha produces several pro-inflammatory effects, including the release of additional cytokines, the activation of additional T cells, leading to a feed-forward pro-inflammatory cycle. Given its central role in pro-inflammatory processes, inhibition of TNF alpha is very effective at reducing immune responses. When it comes to the TNF alpha inhibitors, their adverse effects are all quite similar. The two primary adverse effects expected with these agents are due to its immunosuppressive activity, and these are the presence of an opportunistic infection or a secondary tumor. There are a few others, such as the generation of an autoimmune hepatitis condition, the formation of anti-DNA or anti-nuclear antibodies. The presence of these anti-DNA or anti-nuclear antibodies can sometimes mimic lupus, but true clinical lupus is rare following administration of TNF alpha inhibitors. TNF alpha inhibitors can also exacerbate heart failure, cause injection site reactions, and lead to GI ulcers and perforations. Finally, one of their adverse effects that can be problematic in terms of their treatment are the formation of anti-drug antibodies. Anti-drug antibodies are the body generating antibodies to the treatment TNF alpha inhibitor antibodies. This can cause several things, first of which is a loss of response. If the anti-TNF alpha antibodies are being blocked by anti-drug antibodies, the level of response will be reduced. This can also result in a decrease in serum drug levels, as well as an increased risk of adverse effects from the TNF alpha inhibitor in general. Luckily, the production of these anti-drug antibodies, or ADAs, can be reduced with regular dosing of the TNF alpha inhibitor or by co-administration of an immunosuppressant. That's what's illustrated in the figure here. When there's interrupted treatment with the TNF alpha inhibitor infliximab, upwards of 40% of individuals developed anti-drug antibodies. When interrupted treatment was combined with an immunosuppressive agent, that reduced anti-drug antibodies to 15%. When the agent was given by continuous treatment, that further reduced anti-drug antibodies, and the combination of continuous treatment with an immunosuppressant reduced the anti-drug antibody production close to 5%. Another way to overcome anti-drug antibody formation is by dose escalation or by switching to a different drug or drug class. Now I will talk about some of the specific TNF alpha inhibitors. The first two, etanercept and certolizumab, are what are considered fusion protein or antibody fragment, respectively. Etanercept is an anti-TNF alpha fusion protein. It is comprised of the TNF receptor linked to a small portion of human IgG antibody. It is able to bind and inhibit the activity of soluble TNF alpha in disease states including psoriasis and rheumatoid arthritis. Etanercept does have potential teratogenic activity and therefore would not be the TNF alpha inhibitor of choice in pregnancy. The biologic originator is Enbrel, and Erelzi is a biosimilar. Certolizumab, on the other hand, is an antibody fragment, in this case a humanized antibody fragment, given its zumab suffix, and it's conjugated to polyethylene glycol, or PEG, which increases its solubility and its circulation time in the body. Because it is an antibody fragment, it is much less likely to produce anti-drug antibodies as a result of treatment. Certolizumab is used in psoriatic arthritis, rheumatoid arthritis, and Crohn's disease. Some of the more common and well-known anti-TNF alpha antibodies are listed here, and these include infliximab and adalimumab. Infliximab, again given the suffix imab, is a chimeric human-mouse anti-TNF alpha antibody. It binds TNF alpha and decreases TNF alpha-mediated signaling. As a chimeric antibody, it is the most closely associated with anti-drug antibodies, and these can occur in upwards of 60% of patients. The originator was Remicade, and Inflectra and Renflexis are both biosimilar agents. Adalimumab, or Humira, is probably the most well-known anti-TNF alpha biologic. Given the suffix umab, you can tell that it is a fully human anti-TNF alpha antibody, and therefore is less likely to produce anti-drug antibodies. Some of the biosimilar trade names for adalimumab are Amjevita and Celtezo. Finally, there's golimumab, or Simponi, which is also a fully human anti-TNF alpha antibody. Of the anti-TNF alpha agents, adalimumab is used for by far the most conditions, including Crohn's disease, juvenile idiopathic arthritis, psoriasis, psoriatic arthritis, rheumatoid arthritis, ulcerative colitis, and even uveitis, which is inflammation of the eye. In the next bit of this video, I will start to discuss some of the non-TNF alpha inhibitors. Most of these inhibit interleukin cytokines like IL-6 or IL-23, but there are also other activities I will mention. Probably the most common of the non-TNF alpha inhibitors is Tosalisimab or Actemra. Tosalisimab, again as the suffix zumab suggests, is a humanized antibody against the interleukin 6 receptor. Tosalisimab binds to both soluble and membrane-bound interleukin 6 receptors and prevents it from interacting with its cytokine interleukin 6. This inhibits IL-6 mediated signaling primarily on B cells, which decreases B cell activation, decreases antibody production, and therefore the autoimmune response. It is indicated in rheumatoid arthritis, and in terms of its pharmacokinetics, even though it itself is not metabolized by cytochrome P450 enzymes, it has been shown to increase the activity of some P450s, and therefore drug-drug interactions should be monitored. Its primary toxicities include those caused by immunosuppression, such as opportunistic infection or secondary tumor. It can also lead to some hepatotoxicity and a rare but possible demyelinating disease. There is another similar molecule called sarilumab that was recently FDA approved also for rheumatoid arthritis that also inhibits interleukin 6 activity. Other anti-interleukin antibodies include ustekinumab, anakinra, and secukinumab. Ustekinumab, or Stelara, inhibits interleukin 23, which is another pro-inflammatory cytokine. This decreases activation of both natural killer cells and T cells, as well as inhibits subsequent cytokine production and release. It is used in Crohn's disease, psoriasis, and psoriatic arthritis. In terms of its adverse effects, ustekinumab can cause a rare but serious leukoencephalopathy that fortunately is reversible on drug discontinuation. Next, we have anakinra, which is an antagonist or a blocker of the interleukin 1 receptor. Anakinra is a biologic molecule. It is a recombinant protein and is indicated for rheumatoid arthritis. Finally, secukinumab, or Cosentyx, inhibits interleukin 17, which is another pro-inflammatory cytokine. Secukinumab is also similar to ixekizumab, or Taltz, and brodalumab, or Silic. Of those three agents, brodalumab is the most problematic because it has a monitoring program for the presence of suicidal ideation or behavior. All three are used in the treatment of psoriasis. Finally, there are three additional biologic agents that do not target TNF alpha or one of the interleukins. The first is rituximab. Rituximab binds to a cell surface protein that is found on B cells called CD20. As an anti-CD20 antibody, it targets B cells for depletion by both complement-dependent and complement-independent cytotoxicity. It is indicated for rheumatoid arthritis. Next is belimumab, or Benlysta. This is an antibody that targets a protein called BLyS, which is a protein that activates or stimulates B cells. By inhibiting a protein central to the activation of B cells do not survive and produce antibodies at normal rates. Belimumab is indicated for lupus. Finally, we have abatacept, or Orencia. Given the suffix sept, you can infer that abatacept is a fusion protein. And in this case, abatacept is a fusion protein that binds to a molecule on the surface of dendritic cells or antigen-presenting cells called CD8086. This is a molecule that is required for T cell activation. So by inhibiting CD8086, T cell activation is prevented and the immune process is stopped. Abatacept is indicated for rheumatoid arthritis, and as a strong immunosuppressant does carry the possible effect of an opportunistic infection or a secondary tumor.